So I have no conflict of interest to disclose. So briefly, the outline of this presentation will be that first I will introduce the MEN syndromes, and I will be talking a little bit about the MENX syndrome in the rat that led us to the discovery of P27 as a new tumor susceptibility gene. Then I will talk a little bit about P27 function, and I will focus actually mainly on the MEN4 syndrome in humans. So I hope I'm not disappointing anyone, but I won't be really talking much about the rats. And then I will go into detail on the characterization of some of the P27 mutations. So as you, I'm sure all know, multiple endocrine neoplasias in humans are defined, or a patient is an MEN patient if it has two tumors or tumors in at least two endocrine glands. These syndromes are rare, relatively rare, so the prevalence is about 1 in 30,000, and they have an autosomal dominant inheritance. So when we started our studies in rats, and I'll show you in a minute, so the stand of these MEN syndromes was that there were two syndromes, very well characterized already at the genetic level, so the genes responsible were already known, and they were the MEN1 and MEN2 syndromes. Caused by the inactivation of a protein called menin for the MEN1, which is a tumor suppressor gene, or the activation of the proto-oncogene RET for the MEN2 syndrome. And this is just a very brief summary of the phenotype of these two syndromes. About 10 years ago, we noticed that there was a rat strain that started to develop multiple neuroendocrine tumors, and the phenotype actually of these rats was overlapping both human syndromes, so both the MEN1 and MEN2, and I'll show you in a minute what it is, the phenotype of these rats, and that's why actually it was called MENX, because at the time before actually I moved to Munich, they had already given the name to this rat strain. So what is important about these rats is that this was a spontaneous mutation, so they were not genetically manipulated in any ways. And also, the rats we usually work with are homozygous mutant for the causative mutation. So in these rats, this MENX syndrome is inherited as an autosomal recessive disease. So this is briefly the phenotype of the rats. So you can see here the different organs. If I move the arrow, you see it? Yes. That are affected, so the pituitary, the thyroid, endocrine pancreas, and adrenal gland. And actually, these two tumor types, so pheochromocytoma and these multifocal anterior pituitary adenomas, they occur in all animals. So the penetrance is 100%. And so now if we go back to the scheme of the MEN syndromes, now I added the MENX rat syndrome with the tumors that the rats develop in their frequency in the affected animals. So now, of course, the question was, let's try to identify the gene responsible for this disease, and was not MEN1 and was not RET. This was done by classical linkage analysis. So to make a long story short, we mapped the disease locus to a small enough region that we could really look for candidate genes. And one of these candidate genes was the CDKN1B gene that encodes for the P27 cyclin inhibitor. Then we sequenced this gene and we found a mutation that I don't show you, but we did the analysis on more than hundreds of animals, and in all cases it's segregated with the disease. So we're pretty certain that this is the causative mutation, and is a tandem duplication in exon 2 of P27. Thank goodness P27 is a very small gene, so it only has 2 coding exons. And because of this mutation, you can see here that the protein has actually different C-terminus compared to the wild-type P27 protein starting from the site of this tandem duplication. And because of this very strange C-terminus, it doesn't have any homology to anything, so it's just a bunch of amino acids all together. This protein, the mutant protein, is very unstable, so we did also in vitro studies to demonstrate that, so it's degraded very fast, so that if you look actually in vivo at the rat tumor tissues, you see that there is very little P27, and in some tissues there is no staining at all. So this mutation behaves in vivo as a loss of function, although it's not really a knockout. So a few words about P27, as already indicated, is a member of a family of cyclin-Cdk inhibitors, and mainly binds, as you can see here below, to these complexes, so cyclin E, CDK2 or cyclin A, CDK2, and it mainly regulates the transition from the G1 to the S phase of the cell cycle. And when P27 binds to these complexes are inhibited, and so cell cycle is actually, stops in G1. And the story of P27 starts in the lab of Tony Hunter. In 1994, it was isolated by Cornelia Polyak. And because when it is overexpressed, it actually blocks cell cycle progression, it was assumed to be a tumor suppressor. But then in the following years, the sequencing of many tumors from different groups, Volgestein's lab and other groups in the US, could not find somatic mutations, or very rarely. There were, I think, 3 somatic mutations that were found in different tumors. Of the few hundreds, I think 200 altogether, they were sequenced. So there was a dogma that actually was valid until a couple of years ago. It was always repeated in every review about P27 that this gene is not somatically mutated or rearranged in tumors. But when they looked at the expression level of P27, they found that it's very often downregulated or lost in tumors. And I will show you that in a minute. And I said that this dogma changed a couple of years ago because there was a study done on using next generation sequencing on quite a good number of cases, as it was yesterday afternoon, also reported that about between 7 to 8% of small intestinal neuroendocrine tumors have actually somatic mutations in P27. So this is what I just told you, that in most tumors, P27 is reduced at the protein level, so there is post-transcriptional regulation, mainly due to proteolysis, and these are the tumors in which it is downregulated or lost, and this really usually associates with poor outcome. So just for the sake of completeness, I don't have the time to go into the details, but besides these cell cycle dependent functions of P27, there are also cell cycle independent functions. I would just mention briefly that P27, so in the last few years, people have concentrated really to figure out what P27 does outside the nucleus. They've found that P27 can stimulate cell migration. Again, I don't go into the details, by interacting with, for instance, Rho A in fibroblast and maybe with other proteins as well. And P27 can also indirectly regulate transcription. So it can interact with transcription factors and co-repressors and repress transcription from specific promoters. So the function of P27 is becoming more complicated than we thought a few years ago. So this briefly, I want to show you the functional domains of P27. Again, this is very simplified to just few key points, and here are the cyclin and CDK binding domain, the domain for binding to GRAB2. So P27, as you can see here, interacts with several proteins, and actually the list is now longer. But again, I want to point out just these functional domains because we will come back to those. And also this nuclear localization signal that allows P27 to be shuttled into the nucleus. So I want to stress again that the function as a cell cycle inhibitor of P27 takes place into the nucleus. So if it cannot go there, it cannot work as a cell cycle inhibitor. So I told you that by studying these MENX rats, we identified this germline mutation in P27, and so then we asked the question whether mutations in P27 could be found also in human patients and having multiple endocrine tumors. There was, at the time, I just showed you that there were these two syndromes, MEN1 and MEN2, but patients with an MEN1-like phenotype, in about 30% of cases, didn't show, or people could not find mutations in MEN1 in these patients, so we postulated that maybe they could have a mutation in P27. So we selected, we were lucky that through collaborations we were able to obtain a few patients having multiple neuroendocrine tumors but no mutations in the predisposing genes. And in one of these patients, we hit the jackpot, let's say. So we found this germline mutation that was truncating mutation, relatively early in the protein, as you can see here at codon 76. As you may remember from the slide I showed you earlier, so the full-length protein is 198 amino acids. So this is relatively early in the protein. I will show you a scheme. This is just to say that the patient had acromegaly and primary hyperparathyroidism relatively young. And this is how the protein structure would be, the predicted protein structure based on the mutation we found. And through, again, a collaboration with the endocrinologist there and with a lot of patients, we were able to get some samples from also other family members. So this is our proband with the mutation positive, with this, with acromegaly, primary hyperparathyroidism, and we were able to identify a mutation in a sister that had a renal angiomyolipoma, also a tumor that had been previously associated with the MEN1 syndrome. And then we identified also other family members with a mutation, but with no phenotype at the time of the analysis. So it might very well be that in this family the mutation is not completely penetrant, but I have to say that we couldn't do any follow-up on these patients. They were really uncooperative, and many didn't want even to be tested. So we left it at that. We also did some functional analysis, of course, to figure out, okay, what is it all expressed, this truncated protein, or can we express it in vitro, and what does it do? So for the expression in vitro, it can be expressed, so it's a very short peptide. If one looks at the localization, it stays in the cytoplasm, and this is due to the fact that it no longer has this nuclear localization signal. And at the time, we concentrated for the characterization of these mutants. We were always looking at colony formation, so at eventual inhibition of cell growth by P27. So in comparison with the wild-type P27 construct, the construct expressed in this mutation basically can no longer stop cell growth. So it seemed that this mutation had a functional effect, at least in vitro. When we had access just to one tissue for further analysis, there was the renal angiomyolipoma tissue of the proband sister, and we found no loss of heterozygosity, but no protein expression. So in this tumor, P27 behaves as a canonical tumor suppressor in the sense that it is not present. And I will come back to this issue of the canonical or non-canonical, so this haploinsufficiency concept that was just introduced by the previous speaker. So one mutation doesn't make a syndrome, so we had to wait for a second mutation to be found by an independent study, and this came a year after we published our first mutation. So this was a study done on Dutch patients. And this was, again, a very dramatic genetic mutation. So this was an insertion of 19 nucleotides, which led to a frameshift mutation relatively early in the protein sequence, so at codon 25. So again, one can assume that this is really having an effect on the protein function. The patient presented with cervical carcinoma, pituitary adenoma, this time was an ACTH producing, and primary hyperparathyroidism. This is the predicted sequence of this frameshift, of this mutant P27 with this frameshift that leads to a protein that from this codon has a completely unrelated sequence. And when they looked at the tumor tissue of the patient, they found actually LOH, so the wild-type allele was lost, and they again found no protein expression in the tissue of this patient. So after this report, a new MEN syndrome was added to the OMIM database and was given the name MEN4, which is caused by mutations in CDKN1B gene. And of course, at the time, there were only 2 patients. So these seem to be the common phenotypes, so pituitary adenomas and parathyroid adenomas, as you can see here. So I will show you at the end a summary of all the mutations that have been reported so far. There are only 16, so there are not many. So these mutations are relatively rare. Because of the fact that we have very few mutations, I would like to just show you in a little bit more detail some of the mutations that have been identified and the studies that have been done to try to figure out what these mutations actually do to the protein function. And I will show you a selected number of mutations, which are the ones that we helped characterizing. So this was a mutation that was found in a patient, in an Italian patient. This study was done in collaboration with Maria Chiara Zatelli, and this patient presented with several tumors, so bronchial carcinoid, non-functioning pituitary adenoma, parathyroid adenoma, PTC, and had a missense mutation, codon 69. So again, as opposed to the mutations I just showed you before, this was a mutation for which one had to really study and prove that it was pathogenic, as it always happens with missense mutations. But this mutation occurred in a very interesting amino acid, so it's one of the amino acids that is responsible for the binding to CDK2, this proline 69. So first we checked for the localization. Okay, first we can express it in vitro. Yes, but is expressed at a lower level than wild type, no matter what we did. The localization was also in the nucleus. But when we did pull-down assays to see whether this mutant protein can bind to CDK2, basically the binding is very much reduced compared to the wild type P27. And for colony formation assay, the results were maybe not exactly as we would have expected, but it definitely is less potent than P27 in inhibiting cell growth. We had the fortune actually to be able to study some of the tissues of the patients, so the parathyroid adenoma and the bronchial carcinoids of the patient with the mutation, and we saw no expression of P27 as opposed to other control tissues from patients with no mutations. And the bronchial carcinoid showed LOH at the P27 locus. So this was, again, another example in where the P27 is lost at the protein level in the tissue, so it behaves as a tumor suppressor gene, as one might expect. Then there were mutations also outside of the coding region that were identified, and one was interesting one was identified also in Italy by Gianluca Occhi, and this deletion affects the stop codon of an open reading frame that is upstream of the P27 gene. This mutation was found in an Italian patient with acromegaly and pancreatic endocrine tumors. And because of this mutation that affects the stop codon, as I said, of this open reading frame, now this ORF becomes longer, so it is translated, and the next stop codon is very close to the beginning of the CDKN1B gene. So Gianluca did actually a lot of elegant studies. I have just summarized briefly here that he was able to show that both this elongation of this open reading frame then affects both the transcription and the translation of the CDKN1B gene, so that it is actually less expressed. And this sort of correlates with what he observed in the patient, in the tumor tissue of the patient, that it shows a very reduced P27 expression. There were other changes, other deletions that were found in the 5' UTR of CDKN1B gene, and these are in a region that has a repeat GAGA. They were identified in a collaborative study between Spain and Italy, and then by Maria Chiara again. So functional analyses were done also to study these deletions, and the authors of these papers show that these deletions actually reduce the expression of CDKN1B genes, so they did reported gene assays, and also in the first paper, they saw that it was a reduced P27 expression in the patient's leukocytes. So we come to another mutation that is interesting because of the location of the mutation and the function, the potential function of the mutant protein. So this is a mutation that we identified in collaboration with Maria on a patient that had suspected prolactinoma but developed breast cancer. Then I think that the prolactinoma was not really confirmed. And this mutation occurs in one of the residues that is responsible for the binding to GRB2. And again, by pull-down assays, we could show that the binding is reduced. So the mutant P27 does not bind so efficiently to GRB2 anymore. So in summary, these are all the mutations that have been described so far, as I said, are on R16. And for not all of them, we have the proof that they're really pathogenic. So for instance, these two here, we tried to characterize them, but we couldn't assign a specific phenotype. These mutations, as you can see, can be all sorts of mutations, so missense, nonsense, deletions, and insertions. The most frequent are missense mutations. And by functional characterization of some of these mutants, we have seen that they lead to a reduced amount of transcribed mRNA or protein, increased instability of the protein, this I didn't show you, or impair interaction with protein partners such as CDK2 or GRB2, or abnormal localization of the protein in the cytoplasm where it doesn't suppress cell proliferation. So they seem to be loss of function mutations. This is a summary of not only the mutations, but also the clinical phenotype of the patients. And since it's quite busy, I just summarized here below that the most frequent phenotype is primary hyperparathyroidism in 12 out of 16 patients, then followed by pituitary adenomas, 6 out of 16 patients, and the pituitary adenomas are of different types, so some GH secreting, some ACTH non-functioning. So we have all sorts of tumors. And here I underline the two studies that were published last year because I will come back to this in a second. It seems that every year, maybe 1 or 2 new mutations are identified in patients with these phenotypes. And when I looked at the literature, I noticed that all the probands except one were all females. So again, this is just an observation. I don't know whether this means something or not, but I just wanted to share it with you. So in summary, I showed you that germline mutations in P27 cause MEN4, and this was the formal proof that demonstrated that P27 is actually a tumor suppressor gene. The patients have, of course, the patients are so few that we cannot really be so precise about the tumor spectrum that is associated to these mutations, but the most common phenotype is primary hyperparathyroidism. The second is pituitary adenomas. Mutations can occur all throughout the gene, and they seem to be a loss of function mutations. And again, in the few tissues that people, and we, among others, were able to analyze, it seems that the P27 expression is lost. So it seems that in humans it may work as a canonical tumor suppressor, which is not the case, for instance, in mice, where the reduction of just one allele already predisposes to tumor formation. So the P27 mutations have very low prevalence, but mutant patients are identified pretty much every year, 1 or 2. So I think that if you have patients with multiple neuroendocrine tumors, mainly with an MEN1-like phenotype, but no mutations in MEN1, it is worth to study or screen for mutations P27. The last few seconds, I just want to do a little bit of PR action, telling what we are doing in the lab. So of course, because of this great resource of this animal model that was given to us, we have been trying to exploit it to better understand the function of P27 in tumorigenesis. tumors, but also to study the neuroendocrine tumors that these rats develop. And we were able, over the years, to demonstrate that these tumors resemble their human counterparts, so we can exploit these animals also for translational studies in trying to better understand neuroendocrine tumor genesis, not only in rats, but also in humans. And we can use them for preclinical trials to screen some interesting molecules for their efficacy against these tumors. So with this, I close. I thank, of course, all the people in the lab who have generated some of the knowledge that I showed you today, and the main collaborators throughout Europe who have helped us to get access to the human tissues. So with this, I thank you very much for your attention. Thank you.